dear esteemed
Mm.
members of the nutrition community and fellow enthusiasts of life science worldwide. I'm Dr. Priti Prasad, Program Director at Nutritions, and I am honored to be your host for today's webinar. I extend a warm welcome to each one of you to yet another enriching sessions of our webinar series. At Nutritions, we are dedicated to providing a diverse range of topics and organizing scientific webinars to foster collaboration. It is our privilege to facilitate the gathering of this community to promote the sharing and exchange of knowledge. For exciting news and updates, we encourage you to connect with us on our various social media platforms. Today, we are fortunate to have Dr. Kuldeep Kumar Gupta. He will join us to share his valuable insights in the webinar entitled Regulations of Mycobacterial Polar Growth. Dr. Gupta currently serves as an associate research scientist at Yale University School of Medicines, where he explores the mechanisms governing bacterial phenotypic heterogeneity. With, a nearly, with nearly a decade of experiences in mycobacterial research, Dr. Gupta has studied various aspects such as regulation of biofilm formations, antibiotic resistance, and the cell shape and division regulations in mycobacteria. During the webinar, please feel free to ask your questions in the Q&A sessions, and I will address them at the conclusion of Dr. Gupta's presentations. So, without further delay, I'm delighted to yield the stage to our esteemed speaker, Dr. Kuldeep Kumar Gupta, who will enlighten us about mycobacterial and its phenotypic heterogeneity. Dr. Gupta, the stage is now yours. So please continue. You can continue your presentations. Yes, Doctor.
Yeah.
Yeah.
Thanks, Preeti. Is my screen
Yeah. Is it visible?
visible to?
Yeah. Yeah.
Okay, okay.
Perfect.
Thanks. Yeah.
Thank you, Dr. Gupta.
So yeah, yeah. Hello everyone. And today I'm going to talk about how bacteria grow and divide. And the focus of my talk would be mycobacteria and why we study this bacteria. Why we need to study the growth of this bacteria is because this bacteria causes tuberculosis, TB, and TB is the number one killer of humankind when it comes to infectious diseases. More people die of tuberculosis caused by Mycobacterium tuberculosis than any other infectious pathogen, be it HIV, AIDS, or malaria, or COVID. And even though the treatment for tuberculosis exists, but it takes 6 months to 2 years to treat it. And given the rise of antibiotic resistance, we still so we need to find out new targets for TB treatment so that uh, we can contain the TB threat to public health and also reduce the treatment duration below 6 months. So to do that, we need to know how Mycobacterium tuberculosis grows and divides. So Uh, so regular bacteria like Escherichia coli and Bacillus subtilis, when they grow symmetrically. So they add new cell material in the center of their body, cell body, and then upon division, they produce two daughter cells of equal cell length. But when it comes to Mycobacterium, they grow unevenly. They grow faster at one pole and slower at another pole, and cell division produces two daughter cells of differing cell length. One cell is small, other cell is large. The larger cell, or which is also called fast growing cell, is more sensitive to cell wall targeting antibiotics like ampicillin, while the smaller cell is more sensitive to rifampicin. So now we can see that a cell division in mycobacteria produces a clonal population, but this clonal population is phenotypically heterogeneous. So we wanted to figure out why this phenotypic heterogeneity arises in mycobacteria. So we figured out that there is a gene called LAMA, which stands for loss of asymmetry mutant A. And if we delete this gene, the asymmetric cell division in mycobacteria becomes symmetric. So after the deletion of llama from the genome, what we see that asymmetric cell division, a llama knockout mycobacteria gives rise to daughter cells which are of same length, and more noticeably their sensitivity to all the drugs is same, which makes targeting llama a very suitable candidate for drug discovery. But we do not know how llama functions, how it determines or controls the asymmetric growth. To do that, figure we did a pull down assay in which in this assay we try to figure out the interacting protein of llama to figure out llama's function, and we found out a protein of unknown function called MSMAC0317, and we renamed this protein as polar growth factor A, which we'll come to know later on in the talk why we called it polar growth factor A. So when we started characterizing this protein, we did some bioinformatics and found that this protein is only found in bacteria, which are closely related to mycobacteria. And this protein has a pore-like structure, which means it is might be involved in transport of something, which we still do not know when I started this project. And so first we figured out whether this protein is essential in mycobacteria or not. The way we figure out something is essential or not is to replace it from the genome or delete it from the genome. If a gene or protein is essential, and if we delete it, the bacteria will die. But if it is not, so bacteria will survive. So this is what we did, something called allele swapping or gene swapping. And when we swapped the gene or the deleted the gene in the genome, we found that mycobacteria did not survive. So which goes on to show that the gene is essential. And using microscopy, we also showed that. If you see this, uh, in this time-lapse video, the bacteria are growing happily. And when I delete the gene of interest, that is PGFA, or deplete is using transcriptional, you can see that bacteria eventually stop growing. And if we compare it to the video on the left, here you can see bacteria clearly dying in the absence of PGFA. So we wanted to know the localization of this protein. So we tagged it with fluorescence protein and did microscopy. And we found out that this protein localizes to poles and septum. And here again, you can see we tagged the protein with red fluorescence protein, and you can see that the protein is at the tips of the, and as well as the division site, which is also called as septum. So in these images, you can see we track the localization of the protein throughout the cell cycle. And you can see like FTSZ is cell division marker, which always localizes in the center. So this yellow thing here means that it's always localizing in the center. WAC31 is a polar growth factor. It is always at the poles. So here it's localizing it to the edges of this chymograph. And our protein, what we found, that is it's localizing to both edges as well as the septum. So we think it is involved in polar growth as well as cell division. So the way bacteria were dying, if you see here, they were dying a very violent that their cell wall was popping out. So we hypothesized that their cell wall might be damaged. We used a technique called cryo-electron microscopy, and to image the cell wall, you can see that in wild type bacteria, outer membrane and plasma membrane are intact, while I deplete the protein, the when I deplete the PGFA protein from mycobacteria, the outer membrane and plasma membrane are completely gone. Here are some more detailed images where you can see the damaged cell wall and cell poles. So based on the studies, we concluded that polar growth factor A of PGFA is essential in mycobacteria. It localizes to periplasm and old pole or growing pole, and its depletion causes cell reductive cell division, and which eventually stops polar growth and kills the cell, and it is needed for maintenance of cell envelope integrity. So then, since we had shown that this protein localizes to old pole, so we wanted to see the dependence between what is the relationship between PGFA and llama, because they interact with each other. So here you can see that in wild type background, PGFA is localizing to growing pole here and in the center. And when we knock out llama, so protein does not localize to the pole, and when in the complementary strain, its original position is restored. So as you can see here, so it means there is connection between llama, which uh, and PGF localizes to the whole growing pole in a llama dependent manner. And when in a llama knockout, when we overexpress PGFA, its growth is restored. So as you can see here, this is wild type. In llama knockout, polar growth is reduced, but in when llama knockout background, when overexpress PGFA, the polar growth is restored. We then carried out uh, another cross-linking studies and found that our protein interacts with MMPL3. MMPL3 is responsible for uh, synthesis of mycobacterial membrane. So this is mycobacterial cell wall, this is plasma membrane, this is peptidoglycan, this is arabinogalactan, and this is mycolic acids. So mycolic acids are made from TMM and TDMs, which are the monomers, uh, which stand for trihalose monomycolis and dimycolis. So MMPL3 flips TMM and TDMs from the cytosol into the periplasm. So this is what we know. So this blue structure is MMPL3. It flips TMMs from the cytosol to the periplasm, and then somehow this M, TMM is incorporated into mycomembrane through a protein of unknown function and unidentified. So our hypothesis was that PGFA is interacting with MMPL3 and helps in the transport of TMM. If that is the case, then uh, our protein like PGFA should interact with both MMPL3 and trihalose monomycolates, and this should have right topology. It should be essential for outer membrane construction, and its deflation should phenocopy MMPL3 deflation. So here in these videos, you can see that when we deplete MMPL3 and PGFA, their phenotype is essentially the same. Cells stop elongating and eventually pop out, as you can see here. And then using pull down assays, we showed that PGFA and MMPL3 interact with each other, and we also like isolated TMM and TDMs, and we found that when we deplete PGFA, the ratio of TMM and TDMs changes, which shows that PGFA is involved in the regulation of involved in the transfer of trihalose monomycolate. And we also showed using pull down assay and UV probes and cross-linking that PGFA also interacts with trihalose monomycolates. So Corynebacteria are close cousin or related of a relative of Mycobacteria, and so PGFA has a ortholog or homolog in Corynebacteria, and that is involved in LM-LAM synthesis. So we wanted to see if PGFA also regulates LM-LAM synthesis, and what we found, in fact, PGFA deletion does not regulate LM-LAM synthesis. Rather, it only regulates the level of their synthesis. In PGFA depletion, the level of LM-LAM goes up, and in PGFA overexpression, level of LAM goes down. And when we put the gene NCGL from Corynebacteria and Mycobacteria, it cannot substitute its function. And bioinformatic analysis tells that this sequence identity between NCGL 2760 and MSMUG 0317 is pretty less. And meanwhile, while we were doing this work, another group reported that both PGFA and MMPL3 are highly sensitive drug targets, and this is what we show, and our result also showed. Uh, so in the absence of llama, the PGFA expression goes down. So, and same is true for MMPL3 in wild type. We can see that MMPL3 in PGFA localized at growing pole, in llama knockout at growing pole, their localization is gone, and in the llama complement, the localization comes back again. And this is, you can see that their localization mirrors localization pattern of MMPL3 and PGFA is similar. So then we performed a TNC analysis and found out that in llama knockout background, the levels of uh, SACTI and protein involved in LM-LAM synthesis goes up. So this goes on to show that that llama regulates polar growth in mycobacteria through PGFA levels. And when in llama knockout background we overexpress PGFA, we can see the restoration of polar growth. But when we overexpress MMPL3, that is not the case. So which goes on to show that the polar growth in llama knockout is restored by PGFA, and PGFA is sufficient and necessary for polar growth in mycobacteria. So this is our model that MMPL3 flips TMM from the cytosol, and PGFA, this protein in green, transfer it to my antigen 85 complex, which incorporate into mycolic acid to make mycomembrane, and simultaneously regulating the LM-LAM synthesis. So this our conclusion of our study is that polar growth factor A is an essential periplasmic protein which localizes to old pole in a llama-dependent manner, interacts with MMPL3 and regulates TMM trafficking, and its presence is needed for trafficking of TMM to form mycobacterial membrane. And in llama knockout cells, uh, there is low PGFA, and because of which there is low membrane synthesis. And so, yeah, that is why, like, what we have discovered is a very essential protein which can be targeted for treatment of tuberculosis. So I'd like to acknowledge my lab members and funding agencies and collaborators. Thank you.
Thank you, Dr. Gupta. It's a very short and very informative information about Mycobacterium. And yeah, uh, we have a couple of questions for our from our participants. That
Hmm.
are there any known genetic mutations in PGFA genes that affect polar cell divisions? So, any such kind of research have you done? So, any
Yeah,
genetic
yeah.
specific mutations in the PGFA genes?
Yeah, we have done such mutations, and if we do the mutations and put the mutated copy back in, so it affects the polar growth. So bacteria become even more shorter.
So, there is any
And these,
specific reason or specific loci for that?
yeah, so the position in the gene is the amino acid residue 381.
Okay.
It's the threonine. If we mutate to alanine, the
Mhm.
protein becomes less functional and cell size reduces.
Okay. So the next question is that how do environmental factors such as nutrient availability or host immune responses is any
Hmm.
affects the regulations of polar cell division in mycobacteria?
Definitely, if there is less nutrient, then the growth is slowed down. And same even goes for host factors. In fact, host factors amplify the heterogeneity in mycobacteria. And this is still like an ongoing study, and uh, there is a group in Basel which studies that. Let me just recall the name. They have a very good paper published on effect of host factors on mycobacterial
Okay.
heterogeneity. It's published. It came in 2015 in
So it means
Mol Cell.
then, so it means they have a some certain importance role for the environmental
Yeah,
factors for the microbial growth. Yeah.
yeah.
So the thing is that what does mean by the old pole and new pole? How it is defined in the microbial
Okay,
body? So what is means
yeah,
what is mean by the new pole and old pole?
yeah. Here,
Yes.
can you, if you see here, sorry, can you see the slide?
Yeah, yeah. It's a visible
Hmm.
deletion of lambda.
Yeah,
Yeah.
yeah. So the pole which grows faster
Okay.
here, you see that the this cell, the cell which is labeled with cell wall antibiotic. So
Hmm.
that's called old pole. So old pole is pole which is existing before the cell division, and the new pole is the pole which is formed after the cell division. So here, let me use pen. Do you see this? This is new pole,
Okay.
and this is old pole. This is old pole.
Means the
So
genetic material or the compositions that is present previously and got from the parent
hmm.
parent cells, then we call it old pole. When it's not, is totally generated anew, it's called a new pole.
Yeah, so old pole is pre-existing, and the new poles are formed after division. So do you see this is called septum?
Okay.
So the poles which are just formed right after cell division, they are called new pole,
New.
and they grow slowly, and old pole grow faster. So old poles are called fast growing poles, and new poles are called a slow growing pole.
Okay, interesting facts. Thank you.
Mm, mm.
So, and the next question is that what is LM and LAMN? I think
Okay,
you,
yeah.
yeah,
Yeah, okay, sir.
yeah, please.
So let me, so yeah, do you see LM stands for lipomenan and lipoarabinomenan. These are glycolipids found in the membrane, outer membrane of the mycobacterial here. So these
Mm.
are
Mm.
lipomenan and yeah, these are just variations of what we see, uh, what we see here, like glucose molecules, like arabinogalactan, peptidoglycan. In the same way, they're made from mannoses.
Okay.
Arabinomenan, when linked with lipomenan, is called lipoarabinomenan.
Means it's a glycolipid that is a compositions of outer
Mm.
membrane of the plasma cell,
Yeah,
isn't it?
exactly, exactly.
Okay. Thank you. Thank you. Thank you.
Mm.
So
Yeah, so
the
here is you
hmm,
can see that
hmm,
how LM-LAM pathway is made. It starts from this phosphatidylinositol, which is small lipid, and gradually, several mannose residues are added. And from this mannose residues, it is transported from cytosol to periplasm. Then another mannose residue is added to form lipomenan. And in lipomenan, if you see this structure, which is called arabinan, so it becomes lipoarabinomenan. And then they are incorporated into outer cell membrane.
okay. So come to the
Okay.
next questions. So
Hmm.
next question is that what is there any implications of PGF factors in case of drug discovery, in a case of therapeutic
Yeah,
potentials? Yeah.
definitely.
Yeah.
Yeah, definitely.
So what is
So if
its future perspectives of the study? Yes.
yeah,
Hmm.
so here, if you see that PGFA, before we started studying it, is called MSMEC 0317. So if
Yes.
what this curve shows that if we deplete this protein by a little amount, say 50% here, so even 30 to 40% of the depletion is level causes cell death. So this is a very essential drug target, unknown drug target. So this is what we have discovered, and we showed that it is involved in mycolic acid synthesis, which is the primary target for the uh, mycobacterial antibiotics. So this, if we target any of these layers, bacteria die very fast, especially the mycolic acid. So yeah,
So this
so
will be the putative targets for tuberculosis, for all disease related to this mycobacteria. So it.
yes, exactly. Because
Okay.
this protein is only found in mycobacteria. So if
Mhm.
we target it, so be it tuberculosis or leprosy or any avian mycobacteria like Mycobacterium avium and other, non-tuberculous mycobacteria. That's the beauty of this gene. It's
Okay.
only found in mycobacteria.
So if we target this particular genes, that we can we definitely we will cover up all these life-threatening diseases like tuberculosis, leprosis, like all kinds of life-threatening diseases.
So, yeah, I mean, it's not like, you just target this protein in bacteria die. So it's like a combination. So this is like already existing drugs. If we can design, if there can be a drug which can specifically target this protein, so it
Okay.
will bring down the duration of TB treatment. So this
Okay.
is something pharma companies, this is in the domain of pharmaceuticals,
Yeah,
not in
yeah,
the domain of
absolutely.
basic research.
And that's what science means, that we didn't work in a single mode. We have to work in a collaborative mode to understand all of these things. Okay. So the mean is that the main conclusion of this study is that you want, if you we want to target the PGFA genes, they definitely we will decrease the making the list number of copies of mycobacteria. In a,
Yes.
in a short
Yeah.
take away note from this
Yeah.
presentations.
Yeah.
Okay.
Yeah.
Yeah. Okay. Thank you, Dr. Gupta.
Yeah.
So, yeah. So with this, I would like to thank Dr. Gupta for sharing his experiences and knowledge with us. And thank you all of you for joining us today. So let's continue our journey of exploration and collaboration. Please invite others to join our vibrant community. Together, we can make a significant impact in the world of science and research. We look forward to seeing you all in our next webinar, where we will feature another exceptional life science leader like Dr. Gupta. So thank you, Dr. Gupta.
Yeah.
Thank you for your time.
Yeah.
Other questions, that
Sure.
can mycobacterium related dental disease be cured?
Mm, well, I've never heard of mycobacterial related dental diseases, but yeah, I mean, since mycobacteria, so whatever antibiotics work for, say, pulmonary tuberculosis, I'm sure they should have some effect on the dental disease. But yeah, I've never heard of dental
Okay, so
diseases.
the
No.
disease that is related with the mycobacteria, it will be definitely be cured by the changing a mutational changes in a or some studies on a PGFA gene. So PGFA genes is a major targets for retargeting the mycobacteria, isn't it, Dr. Gupta?
I mean, PGFA is just one target, which should be, like as I
Yeah,
said,
but it is
it's not in our, yeah.
okay.
It has the promise. It
Yeah,
has the
yeah.
promise to become.
Okay.
Yeah.
Thank you, Dr. Gupta, and thank you all for giving your time. We'll see you again in our next webinar. Thank you all. Thank you, Dr. Gupta.